Good morning, students. I'm Dr. Safaa Khaled, Associate Professor of Hematology and BMT at UT University. Today we will have an important topic, which is myeloproliferative neoplasms. In order to understand myeloproliferative neoplasms, we have to revise the normal physiology of hemopoiesis. So first, what are sites of hemopoiesis throughout stages of development? During fetal life, hemopoiesis occurs in yolk sac, liver, and spleen. During pediatric life, in axial and appendicular skeleton. During adult life, occurs in axial skeleton only. Now, what are stem cells? Stem cells are primitive self-replicate cells that differentiate and become specialized progenitor cells, which form mature blood cells. This process is regulated by growth factors such as interleukins, erythropoietin, thrombopoietin, granulocyte colony-stimulating factor. The normal architecture of bone marrow is 50% hematopoietic tissue and 50% fat. Now, if the bone marrow is overacting, it is hyperplastic. If it is underacting, it is hypoplastic bone marrow. Events in normal hemopoiesis: stem cells divide into myeloid stem cells and lymphoid stem cells. Just remember that all blood cells arise from myeloid stem cells except for lymphocytes. This figure showed normal hematopoiesis. As you see, the pluripotential stem cells gives two stem cells, which is myeloid stem cells and lymphoid stem cells. All blood cells originate from the myeloid stem cell except for lymphocytes. Now, another thing you need to know is the normal adult reference range of a CBC or complete blood. This is a blood picture. We need you to discover the abnormality in this. Now, what's chronic myeloproliferative disorders? You see the history of chronic myeloproliferative disorders. In 1951, William Dameshek noticed uncontrolled tri-lineage myeloproliferation in a group of hematological disorders. He named them chronic myeloproliferative disorders. Then in 1960, the Philadelphia chromosome was discovered in chronic myeloid leukemia. The JAK2 mutation was described in BCR-ABL negative myeloproliferative disorders in 2005. In 2008, the WHO renamed the chronic myeloproliferative disorders as myeloproliferative neoplasms owing to their clonal nature. So, what are myeloproliferative neoplasms? Myeloproliferative neoplasms are clonal disorders originating in multipotential hematopoietic progenitor cells. They are characterized by overproliferation of one or more of myeloid lineage-derived cells that is independent from cytokines with relatively normal differentiation. Just notice that all the physiology which we told before was reversed in myeloproliferative neoplasms. Now, what are common features of myeloproliferative neoplasms? They are acquired hematopoietic stem cell disorders characterized by overproduction of one or more of formed elements of the blood. They could be complicated by bone marrow expansion and extramedullary hematopoiesis that takes place in liver and spleen. Could be complicated by myelofibrosis or transformation to acute leukemia. The other thing is the transition between different phenotypes of myeloproliferative neoplasms could occur. They harbor cytogenetic abnormalities and they could have thrombotic or hemorrhagic death. Now, WHO classification of myeloproliferative neoplasms. The WHO classify myeloproliferative neoplasms into classical myeloproliferative neoplasms, atypical myeloproliferative neoplasms. The classical myeloproliferative neoplasms were subdivided into Philadelphia positive, including chronic myeloid leukemia, Philadelphia negative, including polycythemia vera, essential thrombocythemia, and primary myelofibrosis. The atypical myeloproliferative neoplasms, including chronic neutrophilic leukemia, chronic eosinophilic leukemia, hypereosinophilic syndrome, chronic myelomonocytic leukemia, systemic mastocytosis, and chronic myeloproliferative neoplasms, unclassifiable. In 2016, the systemic mastocytosis is no longer considered a myeloproliferative. What's the pathophysiology of the chronic myeloproliferative neoplasm? In BCR-ABL positive chronic myeloid leukemia, reciprocal translocation between long arms of chromosome 9 and 22 occurs. The genus kinase 2, calreticulin, and myeloproliferative leukemia virus mutations were detected in BCR-ABL negative myeloproliferative neoplasms. However, nearly 10 to 15% of primary myelofibrosis and essential thrombocythemia cases, their mutational analysis is triple negative. This figure shows the BCR-ABL gene in Philadelphia positive myeloproliferative neoplasms, and figure 3 showed percentage of patients with mutation in BCR-ABL negative myeloproliferative neoplasms. And finally, this is my references if you need more reading. Thanks for listening.